So in this video, we are just going to be looking at the structure of the DNA. Okay, I'm just going to be drawing out what we know so far is DNA is a polymer and it's made up of monomers known as DNA nucleotides. And DNA nucleotides are these small molecules. They are made up of a phosphate group, a deoxyribose sugar, which is a type of pentose sugar with 5 carbon, and it also has nitrogenous base. I've simplified the nitrogenous bases over there, and there can be 4 possible nitrogenous bases that can exist, which are nitrogenous base A, adenine, T, thymine, C, cytosine, or G, guanine. When you join these DNA nucleotides together, you can get something called DNA molecule, which is the polymer. And that's when it forms the very familiar shape that we know. When I ask my students what is DNA, they always tell me, yeah, DNA is that swirly thing. It's found in the nucleus in our cells, and it does things. It affects our characteristics. It affects how we look like. It affects our appearances. But how though? Why does that swirly thing made up of nucleotides affect how we look? It affects our skin color, our height, our hair type. To a certain extent, it might also be able to affect our personality. But that's also environmental as well. Okay? So, we cannot downplay the effect of DNA on our daily lives. See, here's the thing. All living things have it. We know that it affects our appearances. And as you can see, this otter smelling flowers, quite a cute picture. The otter has DNA and the flowers have DNA as well. So why does the otter look like an otter and why does the flower look like a flower? It's kind of weird, isn't it? Because there's nothing inherently much different about the chemical structure of the DNA in the otter and the DNA in the flowers. Yet they look totally different, as you can see in the picture over here. All right, I just used that as a picture because I just came across this picture online and I thought it was the cutest thing ever. So forgive me for that. All right. Now, before we go into that, we want to talk a little bit about the structure of DNA. A molecule of DNA is basically made out of 2 strands, as you can see here, one in black, one in red. Some students ask, is it really black and red? I'm like, no, it's not. This is just for my illustration purposes. And the shape of DNA is referred to as a double helix. Double because it's made up of 2 strands, helix because it's spiraling along each other. Now, if I were to just basically uncoil and unwind the DNA and just put it side by side, like straightening them and putting it side by side, we will notice that the 2 strands are not directly touching each other actually, okay? But the 2 strands are connected to each other by something called bases. Here is where the purines and pyrimidines have to be discussed a little bit. Now, James Watson and Francis Crick understood the people who conceptualized the shape of DNA. Okay, they did not discover DNA, by the way. I would just like to put it out there. James Watson and Francis Crick. People knew of the existence of DNA, I think, around the 1930s. James Watson and Francis Crick were the people who came up with the structure of DNA, and that is why they won the Nobel Prize in Physiology and Medicine. There is no Nobel Prize for biology, weirdly enough. There is one for chemistry, there is one for physics. We don't have one for biology yet, sadly. Sucks. But anyway, that's just a list of problems which irk me. But anyway, coming back to this, okay. So James Watson and Francis Crick understood that there are 2 types of nitrogenous bases, which are the purines, which are the larger of the nitrogenous base, made up of 2 ring structures, and the pyrimidine, which is a single ring structure. They understood that if one base had purine on one side and the other side also had purine, it would be too close to each other. Also, if one side had pyrimidines and the other side also had pyrimidines, it will be too far for them to form a chemical bond, a bond with each other. Therefore, they understood that if one side has to have purine, the other side has to have pyrimidine. That's when the distance is just right. Purine and purine are too close. Pyrimidine and pyrimidine are too far. But a purine and pyrimidine can form a distance that is just right for them to connect to each other. And one purine base will always pair up with one pyrimidine. And they understood that if there's an adenine on one side, it will always pair up with thymine. And if cytosine is on one side, it will always pair up with guanine. This is universal, and I want you to understand this. No matter whether you're a human, a cactus, an otter, a bacterium, or even an algae just floating in the lake somewhere, your DNA will behave the same way. Adenine will always pair up with thymine, cytosine will always pair up with guanine. There are exceptions, but these are extremely rare exceptions that arise due to mutation. But under normal circumstances, adenine pairs up with thymine and cytosine always pairs up with guanine. And when adenine pairs up with thymine, they will form 2 hydrogen bonds. And when cytosine pairs up with guanine, they will form 3 hydrogen bonds with each other. And I'm just drawing it over there for you to see. This phenomenon is referred to as something called complementary base pairing. Complementary base pairing means the bases have to match each other. A will always want to match up with T, and C will always want to match up with G in these circumstances. For example, let's look at an example. I'm drawing out a strand on the left side where the base sequence is ACGGC. What will be the sequence on the other DNA strand? Obviously, it will be T, G, C, C, and G. And that is referred to as something called complementary base pairing. And I'm also going to draw out the hydrogen bonds to show it to you. As a reminder, A and T will form 2 hydrogen bonds, C and G will form 3 hydrogen bonds. Now, we are not done with DNA yet, okay? We are going to go into the detail of DNA. So what I'm going to do is I'm going to draw out the base sequences, and I'm just going to make it more detailed to show you how the individual nucleotides look like. You can see the individual nucleotides, the sequences are still the same, okay? And I want you to see how the nucleotides are connected to each other. The nucleotides are connected to each other with the purple color line, okay? And when the purple color line forms that, I've highlighted one strand just to show you how one strand looks like in yellow, and I've highlighted the green color strand to show you how the other strand looks like. It connects the nucleotides together. So we know that the DNA strand, which I've just basically circled, it's made up of DNA nucleotides joined together by something called phosphodiester bonds. Phosphodiester bond is a type of covalent bond. And the phosphodiester bond basically links one nucleotide to another nucleotide so that they can form a single DNA strand. Now, you must understand that the phosphodiester bond is between the sugar of one nucleotide and the phosphate group of the other nucleotide. That is important to know. I'm highlighting it, I'm drawing it over there for you to see that. The sugar, which I've highlighted in yellow, okay, and the phosphate group, which I've highlighted in green, so that you can see how the phosphodiester bonds form between one nucleotide and the other nucleotide. That is important because sometimes if they do ask you to draw a DNA strand, you might have to draw the phosphodiester bonds. So the placement of the phosphodiester bonds is quite important. Now, another very interesting thing to also know is both strands are actually anti-parallel to each other. Now, what do I mean by anti-parallel? They basically run in opposite directions. To make it simple, I'm just going to show you a nucleotide on one side and a nucleotide on the other side. Notice their direction. One is sort of facing upwards, and the other one, the phosphate group, is facing downwards. Okay, so just to give you a direction, that's how it kind of looks like. That's where they're facing. So does the DNA actually have directions? Yes, they do. Okay, we usually use something referred to as the 5' and 3' direction. And this is something that confuses a lot of students. They're like, what makes it 5? What makes it 3? Okay, I've gotten a simple way of teaching this. I hope you can follow me for this. All right, the first thing we want to do is at every corner of the DNA molecule, I'm going to just put a single blank space. Okay, you can see the blank space in black that I've just basically written over there. Okay, now what I'm going to do is I'm going to zoom in. Okay, so and we're going to see which side is going to be 5 and which side is going to be 3. So let's zoom in into one corner of the DNA first. Now at this corner, let's label the carbon. Remember, the carbon in the deoxyribose sugar is supposed to be started with carbon number 1, carbon number 2, number 3, number 4, and number 5. And carbon number 5 is the closest to that blank space. So we will put 5' over there. It's as simple as that. That's 5'. And if we were to go to the bottom left corner, we do the same thing again. Let's label the sugar, carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. And we notice that carbon number 3 is now closest to the blank space. And therefore, we just put 3' over there. Simple as that. All right? And because it's anti-parallel, and because the DNA runs in an anti-parallel, which means to say they are running in opposite directions, okay, that side has to be a 3', and the lower right corner has to be a 5'. If you don't believe me, let's check it out. Okay, let's look at the bottom right corner. Let's label the carbon. Carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. And carbon 5 is the closest to the blank space over there. Thus, that will be 5', basically. And of course, on the top right corner, same thing. Carbon number 3 is closest to a blank space, and therefore that is 3'. That's basically it. So this is where we have to ask ourselves the question, why do I even care that the DNA is running in the opposite direction? Does it really matter? Yes, it does. Sadly, I can hear the collective groan from all my students that, oh God, why? But that's just the way it is, okay? So we are going to talk about it a little bit more in detail. So you see, why does it matter? It matters because new nucleotides are always added in a 5' to 3' direction. Now, what do I mean by that? Okay, let's give ourselves a situation. Okay, I'm just drawing out a nucleotide on one side. Okay, just a long strand of nucleotide. Okay, as you can see over there. And I've also labeled them as 5' and 3'. Now, I'm also going to draw out on the other side. You notice that the DNA strand on the other side is quite short. Okay, it's only made up of 3 nucleotides. Now, let's say I want to make that strand longer. Therefore, I would like to add more nucleotides into the strand. So the new nucleotide, does it go to the top portion or does it go to the bottom portion? Remember, new nucleotides are always added in a 5' to 3' direction. Therefore, in this diagram, it will actually be added to the bottom. That's how it works. So it will be added to the 3' direction, and it will just basically keep going on and on and on until the addition of nucleotides stop. That's basically it. To summarize, DNA is basically made up of DNA nucleotides. They are basically joined together by phosphodiester bonds, right, to make a single strand. But DNA is two strands, right? So the two strands are anti-parallel to each other, which means to say that they are running in opposite directions, okay? And I'm also going to label the 5' and 3'. And the two strands are also connected together by hydrogen bonds via complementary base pairing. For example, if on one side it has A, the other side will have T, one side has G, the other side has C, one side has T, the other side has A. And they form hydrogen bonds with each other. I'm so sorry, there's a bit of my G and C is a little bit of an angle, just forgive me for that, okay? And the third thing that we must also know, the two strands coil around each other, forming a double helix. This is in summary the structure of DNA. The highlighted parts that I've highlighted are usually the things that you need to explain in the exam or list in the exam if they ask you to describe the structure of DNA.